This Week in Virology, the podcast about viruses, the kind that make you sick.
From Microbe TV, this is TWiV, This Week in Virology. This is a special episode recorded on May 23rd, 2019. I'm Vincent and you are listening to the podcast all about viruses. Today we're back at Cold Spring Harbor Laboratories. We are at the Retroviruses 2019 meeting. We were here for the first time last year. We must have done something right because we are back, and I'm happy to be back again. And we are going to honor one of the keynote speakers at this meeting, and we've collected him and some of his former people that he's worked with. And so let me introduce them to you. He is from Duke University, Bryan Cullen. Welcome.
Hello, everybody.
And we have three scientists from different stages in Bryan's career to reminisce and hopefully roast. From Fox Chase Cancer Center, Ann Skalka.
Ready for roasting?
Yes. And this is Ann's third TWiV. From the King's College in London, Michael Malim. This is your first time.
It is my first time.
Welcome. And from the Rockefeller University, coming back for a second time, Paul Bieniasz.
Hello, Vincent. Hello, everybody.
And we have an audience, which is very nice. I want to ask you, I was going to say, is it an enthusiastic audience? It sounds like it. I want to ask the audience a few questions before we start talking with Bryan. Who works on something other than a retrovirus? Yeah, a few people here. Yeah. You work on Ebola, right? All right. Who works on lentiviruses? Raise your hand. Okay. How about alpha retroviruses? Few. Beta retroviruses? At least you know your classifications.
Barely.
Gamma retroviruses. We got some of those. And finally, delta retroviruses. 1, 2, 3 people. And foamy viruses.
No.
All right. That's cool. I did that last year. I wanted to see again who's here. So of those of you not doing retroviruses, what are you doing? Shout it out. Okay. All right. So at the end, you're going to stay. We're going to finish at 2 exactly. Okay. You see this thing in the front? This is a T-shirt. This is a special edition TWiV World Tour T-shirt. It's designed by Steampunk Phage. You can find them online if you want to buy one. On the back is a list of all the tours we did last year, and it includes Cold Spring Harbor 2018 retrovirus meeting. So I'll toss these out at the end. So if you really like TWiV, who likes TWiV? Cool. I do too. Yes. All right. So let's talk a little bit about Bryan. I want to explore your history for a bit. And I do want to start by saying it's Bryan, right?
Yes, and if everybody could remember that, I'd appreciate it.
I heard Bryan give a talk a couple of weeks ago in Rotterdam, and he started by saying it's Bryan. Exactly that. All right, where are you from originally?
I was born in a city called Bradford, which is in Yorkshire in northern England. And
you went to early years of schooling there, I presume?
Yeah, until I was 7, and then I lived in Germany for 5 years, which accounts for the lederhosen that I was wearing in that picture. Went back to the UK when I was 12, and then stayed there through undergrad and a master's degree before moving to the States.
Where did you do your undergrad?
Warwick University, and then a master's at University of Birmingham.
So were you a science major?
Yeah, it was biological sciences at Warwick and virology, actually focusing on herpes viruses in my master's.
Mm-hmm. And do you know where your interest in science came from?
I think reading science fiction books originally.
So you have a favorite science fiction author?
That's evolved over the years, but yeah. Science fiction fantasy. Guy Gavriel Kay is a big favorite of mine. Dave Duncan for lighter type fiction. Those days I would have said Arthur C. Clarke, Isaac Asimov, Ray Bradbury, the sort of superstars from those days.
Okay. Science fiction. That's what we do, right?
Well, we try to make it less fiction and more science. Yeah.
Speak for yourself.
I think the public, in part, thinks we do science fiction because when they like science fiction movies, but if you make a real science movie, they don't like it as much, right? So we got you through a master's in the UK. Then what did you do next?
Well, my parents had moved to the US when I was an undergrad, and so rather than staying on and doing a PhD in the UK, I thought I'd spend a year or two in the US. And it turned out I hadn't read the fine print because if you leave the UK and live overseas for a couple of years, you no longer count as a resident, and therefore you can't get a grant to do a PhD from the UK government. So the option of going back and doing a PhD there sort of evaporated. And so worked as a tech for a number of years, and then when I was at Roche, went on to do a PhD, which what was then UMDNJ and is now part of Rutgers in New Jersey.
What lab were you in Rutgers?
It was sort of a unique circumstance because my mentor at the time, Grace Jew, became an adjunct professor. And so I was actually able to do my research in the laboratory that I'd been working as a technician, which was kind of unfair because I already knew how to do everything I needed to do before I even started doing the PhD. And so I used to spend a lot of time driving back and forth from Nutley to Newark to do the classes and labs and stuff, but the research was all done at Roche.
And what was that on?
That was on retroviruses, actually. Grace, who had been a postdoc in Ann's lab and was very interested at that time in avian retroviruses. And so working with Grace and also with Ann and Richard Katz and others in Ann's lab, we published a number of papers. I think I had about 7 out of the PhD that were all dealing with avian leukemia virus or sarcoma virus.
All right. So then after your PhD at Rutgers/Roche, what did you do next?
Well, the whole, so Roche essentially sponsored my PhD. They actually paid me full salary while I was a graduate student. And so they kept me on as a lab head. So I never did a postdoc, which is kind of unusual then and almost unheard of now, I think.
So you stayed out at Roche for a while.
From 1984 till I got an offer from Duke in 1987 for a Howard Hughes investigator slot.
All right. And you've been there ever since?
Been there ever since.
Working on viruses, which seems to have started way back in the UK, right?
Yeah, I've always been interested in viruses, and I think, the thing that attracted me a lot to viruses, particularly early on, was once molecular biology got going, around 1980, 81 or something like that, the ability to make mutants and analyze phenotypes was so much easier in a small virus like avian leukemia virus or HIV than it was in a herpes virus, which, is extremely difficult to manipulate.
So what years were you at Roche then, from technician through the end?
It was actually from I started there in December of 1974, and I left there in October of 1987. So it's quite a long time.
I asked because I interviewed with Aaron Shatkin, and Ann came to my seminar. I remember that very well. I don't remember you coming to it, though.
Wow. I don't remember whether I was there either.
But I would have remembered you, yeah, for sure.
I don't know.
Clearly, it was a memorable seminar. Obviously,
I certainly interacted with Aaron a lot. He was a great guy.
He was actually my neighbor. I moved to a nearby town not too long ago, and I used to see him jogging by on Saturdays, and he lived right around the corner from me. Yeah. So I did not go to his laboratory, but it was an interesting visit. So Ann, you're the earliest person of this group to have contact with Bryan. How did you know him? Tell us about that.
Well, I knew him because he worked for, I think it was 3 different people as a technician in the department that I was in. And Bryan was always very interested in new technologies and cloning and all of that stuff. And of course, that was what we were doing at the time because I was a phage person and then a retrovirus person. And so I knew we got together quite a bit. And he was one of the team. I mean, he was essentially like another postdoc, or he was that capable.
Because looking at PubMed, you have a few papers together, right? 4 or 5 papers.
We have 3 papers together. I think we did a review together, and we did a book together. Yeah.
But this was a casual thing because you were working for someone else. Is that right?
Yeah, well, I was working for Grace Dew, but Grace Dew was an ex-postdoc of Ann's, and they maintained a close relationship. And so that was in part why I was working with people, with Ann and with people in Ann's lab.
Yeah.
Do you remember anything about him from those days?
I do, yes. So the thing that was most striking about Bryan at that time was that he had golden hands. Everything he did seemed to work. And I mean, he was really technically so skilled, and it really ticked all the other postdocs off because he'd leave at 5:00 every day. And it didn't matter. I mean, he got his work done. It was amazing. And so what else about Bryan? Well, he was always very self-assured.
Is that a polite way of putting it?
I have a picture of Bryan during those times. Can we have that slide?
Working on it.
He's trying. So you can see we were both a little bit thinner at the time. Check out the bell bottom. And look at that position.
Wow.
That was taken at about 1980, I think, in a group shot. Can I have the next? Want to ticket? Yep. That was at the Roche Institute, and that was taken right in this front of this door here in a group shot. That was a lovely time. It was a time that we were working in a research institution and could do anything we wanted and didn't have to write a grant, and had wonderful facilities. It was really a Camelot. But of course, like all things, it actually eventually died. Can I have the next slide, please? I always thought he looked a little bit like a beetle, don't you think?
As long as it's not Ringo.
No. Anyway, no lederhosen, but that was a younger Bryan.
You remember? Do you still have those pants?
I'm wearing the same shirt.
Yeah. Do you have fond memories of those years?
You know, it was an incredible learning period. You know, I'd done some technology, you know, as a master's student. You don't do very much as a bachelor's student, of course. And, you know, being in 3 different labs that had 3 entirely different research programs. One was developmental biology, one was like gene regulation, and then the third one was a virology lab. I picked up a huge number of techniques, and I really like to learn new tricks and then try to apply them in a different context. You know, I've always thought that the best way to move ahead is to take something from another area and move it into your own area and do something that nobody in your field has done yet because they hadn't been aware of that technical skill being movable. And so, yeah, I mean, it was a huge learning experience, very positive.
And Ann says you were fascinated with technologies. Is that correct?
I was extremely fascinated with cloning, and that was for a while. I was the person at Roche who could express things. So I built a whole bunch of expression vectors when you couldn't just pick up a catalog and get one off from somewhere and got. And of course, there was no internet or anything like that. So there was I interacted with a lot of people, and that's really what attracted, I think, the company to me when they hired me out of the institute into the company, because they were interested in expressing interferons and interleukins and things like that at usable levels. And that was something I spent a lot of time thinking about. And that stood me in good stead later when I was trying to study gene regulation in HIV.
So when's the last time you picked up a pipetman?
You know, the nice thing about when I moved to Duke was because I was a Howard Hughes, I didn't have to write any grants. And so I was able to stay in the lab a lot longer than most assistant professors do. So I think I probably did the last figure in a paper that I did was probably around 97, something like that. Did I have any figures in any of your papers, Paul?
No. So maybe I need to refresh your memory a little bit. So I arrived in 96. You didn't do any experiments after that. But it's clear you had some attachment to the pipettes, because whenever there was a reagent request, Bryan would actually go to the fridge and pipette an aliquot of plasmid into a tube and send it off. So
I consider that an experiment.
So do you miss doing experiments, or are you happy not to do it?
You know, what happens is I had a certain skill set, like I could do S1 nuclease assays and immunofluorescence and things like that. But the technology's moved on, and then that skill set wasn't really applicable anymore. And, I would have had to do what I had done previously, which is to, learn new tricks. And that became less appealing over time. So, as the lab grew and you get more responsibilities and people invite you for seminars and you get more papers sent to you and grants sent to you, you have less and less time. So that's my excuse anyway.
Okay. Now, Mike, you arrived at Duke in 87?
87, that's right, yeah.
Were you his first postdoc?
No, I was his second postdoc.
Okay. And what made you decide to go to his lab?
Okay, so I was doing my PhD or a DPhil in England in the mid-80s, and during that time, it was in biochemistry actually, and during that time it was the isolation discovery of HIV, its emergence as a major disease-causing agent, and I decided I wanted to go to the States for a couple of years to work on HIV. I would switch fields, work on HIV, and so I wrote to a whole load of different virology labs in the US, and eventually picked Bryan's lab, even though his was one of the smaller groups. But I was inspired by two papers he had published. One was an ALV paper we talked about the other day, the transcriptional overlap interference paper when he was at Roche, and then his single author cell paper came out in 86, I believe. And so I actually interviewed with him at Roche, and then while I was trying to make my mind up about which lab to go to, he called one day and said, well, actually, I'm going to move to North Carolina. How do you fancy North Carolina? I decided to go with that, and it was definitely the right decision.
So, Bryan, did you pick up on HIV immediately after it was initially discovered?
I actually was given a very good opportunity. So if you go back to when HIV was sequenced in like 85. Nobody had HIV-1 proviral clones, and most people were scared to death of working with HIV at that time. And so Hoffman or Roche bought a proviral clone from HXP2 from NIH, or licensed it or whatever they did. And so I actually had a full-length HIV-1 proviral clone in my hand. And I had all these years of experience of working with avian leukemia virus, a retrovirus, right? So I figured I can just take my ALV experience and try to apply it to HIV-1. And so I'd been particularly looking at transcription in avian leukemia virus and in endogenous retroviruses, working with them. And so I knew how to measure transcription accurately, measure RNA accurately, and things like that. So it was a question of just taking the tools I'd already developed and putting them into a new system. And it was really fortunate because, there were only 3 labs doing molecular biology of HIV for a couple of years. There was Hasseltine's lab, Wong-Stiles' lab, and my lab, and that was essentially it in terms of the molecular biology side of the virus. And so there were a lot of opportunities that presented themselves starting in about 1985 that I was able to take advantage of.
So, Mike, when you arrived, HIV was going strong in the lab, right?
Well, we were unpacking boxes. So, but it got going fairly quickly, yeah. And there was an excellent postdoc that was already there, Yogi Hauber, who moved from Roche to Duke with Bryan. And you'd hired Randy Fenrick to set up the lab, and it was the three of us, four of us.
Yeah, the four of us. That was it for a while, and I thought that was great. I mean, you got four people who all know what they're doing, and that's incredible if you're an assistant professor. I mean, to be able, I think that's almost the sine qua non of trying to be a successful assistant professor is getting a couple of people into your lab who actually are capable of moving projects along. And if you can do that, you're so far ahead. I mean, because your hands may be good, but you're one person, and you can't compete if it's just one person. But to be able to have a couple of really good postdocs and a decent technician right from the word go, it was the defining. Gave me the trajectory that I was able to achieve.
What did you work on in the lab?
I actually worked on REV, and nuclear export was the project I worked on to start with. Yeah, in fact, Bryan had not published a paper on REV when I joined the lab, and I think his first task that he gave me was go away and think about an assay that we can measure REV. And we were very fortunate, actually. The first assay we tried, which was just a very simple intron-containing reporter. Bryan previously had made a very, very good antibody against the TAT protein, and if you know about HIV molecular biology, the TAT gene is split with an intron, which is response to REV, and the first exon ends with a stop codon straight in the intron, so you could actually distinguish between the two translated products of TAT with this very good antibody that Bryan had, and so we were able to use radiolabeling and immunoprecipitation to rapidly have a very quick assay for measuring REV function, which gave us a very good competitive position immediately.
Was Bryan working in the lab at the time?
He was, yeah.
Did he give you any help?
Yeah, he used to walk around the lab. He didn't like pipettement very much, so he always used to walk around with this bit of rubber tubing dangling out of his mouth with one of those little thin glass tubes he had, which I've forgotten the name of. So he used to, and then when he got distracted and the phone would go, he'd sort of leave this rather nasty thing lying on his bench. And he did. He did a lot of lab work, actually. I think he taught me how to transfect cells, S1 nuclease protection assays. Yeah.
These were little capillaries that you...
that's it. Yeah, those things.
I used to pull my own capillaries, yes. Yeah. And I used to, pipette P32 labeled probes into the little slots on the top of the gel. So.
And you would seal the ends? To do hybridizations, you could seal the capillaries and put them in water baths.
No, we didn't used to do that. I used to do the capillaries because, the quality of the pipette that we had in those days was not very high, and the sizes were not many available. So to try to get a really tight band in the bottom of the well when you're doing because these were urea gels, so the urea would leach into the well, and then the sample wouldn't go to the bottom, and then you'd end up with this blob instead of a nice band. So that was I used to blow out the well, and then add the blue dyed RNAs, what was left after the S1 protection assay.
Yeah, this is a thing of the era, these capillary tubes, I remember. I remember someone who could put one in between each finger and put reagents to do sequencing, right? We used to do them in these tubes and then seal them all. This was Nigel Godson used to do this and then put them in a water bath. Was he good at troubleshooting?
Well, he was good at causing trouble. Yeah, I mean, as Ann says, I mean, he was extremely good in the lab. Actually, he had very few experimental failures. Yeah, and it's rather frightening to hear that Paul reminding us that he still pipetted the plasmids out when they get sent out. I have to admit, I still do that, which is the only lab work I ever do. Which is
you don't have to.
Well, you said he caused trouble. What kind of trouble did he cause?
Well, it was one thing we used to play. We used to actually we didn't spend time on the internet because there wasn't any. So when we used to chat about science, we used to have we actually wore a lot of them out. We had a Nerf basketball thing on the wall, and we alternated between that and Velcro darts. And we used to spend a lot of time playing these games while we talked about science. And we used to irritate people when they would come into the lab to try and talk to us.
Yeah, Warner Green used to get really annoyed with us, actually.
But no, he didn't cause too much trouble.
Did you ever have disagreements of any sort?
I think we did, yeah. Twice.
Scientific ones, I assume, right?
Yeah, I think so. Yeah.
Don't
I
remember.
any of them.
I thought everything was, hunky-dory.
I do remember one. We did scream at each other down the corridor once over the value of reporter gene assays versus actually measuring RNA. And I don't think we spoke to each other for a few days after that one.
Which side were you on?
I was telling you to measure the RNA.
Yeah, okay.
Is that the right side?
Probably. Although they're not mutually exclusive.
So since the lab was relatively new, you probably saw Bryan a lot on a day-to-day basis, right?
We did, yeah. Yeah.
It's not always the case. Probably now you don't see people as much as you did back then.
Nowhere near. Yeah, I mean, in those days I was in the lab a lot and very interactive and very hands-on as well in terms of trying to talk to people every day about exactly what they were doing. Probably annoyingly so, but these days I'm much more of a, let them go and see what they can manage and talk to them once a week kind of philosophy.
Now, Paul, you didn't overlap with Mike at all.
No, I think Mike had been gone about 5 years before I showed up.
And why did you choose to come to Bryan's lab?
Well, I guess a couple of things led me to Bryan's lab. So I did my PhD with Myra McClure in London, and the first thing I worked on was trying and failing to identify HIV co-receptors. And one of the sort of classic papers in the field at that time had come from Bryan's lab, identifying the envelope determinants that governed HIV tropism. So I knew of him from that work. Then I started working on foamy viruses for the latter part of my PhD, and I would run into papers from Bryan's lab about transcriptional regulation of foamy virus because he dabbled in it at that point. Basically, everywhere I turned, I couldn't get away from him in a sense. But I don't think I've ever told him this, but he wasn't my first choice of... So when it came to apply for postdoc positions, my father had visited Seattle and told me what a wonderful city it was. So I was really sure I wanted to go and do my postdoc in Seattle. So I wrote letters to Maxine Lineal, Dusty Miller, and Michael Emmerman, who were all at the Fred Hutch at that point. But in those days, of course, you'd actually send a paper message. There wasn't any of this electronic stuff. And apparently, at least a couple of them went by surface mail. So, I'd mailed them, not putting an airmail sticker on. I thought that was assumed by then. But a couple of them didn't get there. And then I think Dusty Miller wasn't so enthusiastic. So I went to my second tier of applications. And it just, as it happened, I had... met Bryan at this meeting that would be 1995. And although, well, let's say everyone in the field knew at that time, Bryan didn't have a reputation for being particularly laid back. Let's put it that way. So I was a little frightened of him. But I actually went up to him and talked to him at this meeting, and he wasn't as bad as people had made out. And during my interactions with literature from his lab, I, actually read the papers and read the methods section, although molecular biology had become established as a, not routine, but fairly easy to do set of techniques by that time. I'd basically learned it myself from these red books and the Bieniasz books in the lab. There weren't any other molecular biologists around in my lab at that time. But reading Bryan's papers, or papers from his lab, I was sort of blown away, not just at the breadth of the topics, but also the sort of the technological sophistication. And sort of thought, if I really want to learn molecular biology, this would be probably a pretty good lab to go to. So I wrote to Bryan, and this time it actually went airmail. And Bryan's also once you get to know him, you'll understand that he's not the sort of person to procrastinate. So within a few days of me sending that mail, he'd spoken to Myra, my boss. He then called me, I think the same day. We had a chat on the phone, and he basically offered me a position over the phone, and there was no reason to say no, basically. So,
do you remember this exchange, Bryan?
Not the part about writing to Seattle, but yeah, I do remember meeting Paul at the meeting, and he seemed very shy, which
that's true. That's true.
Certainly, over
You're
time.
a scary guy.
You were taller than I was, weren't you? And his interaction was very positive, and the questions he asked were very insightful. And then when he talked to me on the phone, I realized he'd actually read some of the stuff I'd written, which I thought was really unusual in an applicant for my lab, because usually you don't know what the hell I've done in the past. So that was, it was a no-brainer, and that was one of the decisions that certainly turned out to be 100% correct, making him that offer.
Had you read his papers before?
Absolutely, I
Yeah.
had, yeah.
Okay.
Classics.
I think there were only about there were a lot fewer at that point.
So, what did you work on when you arrived, Paul?
So in that phone conversation, we even got around to talking about that, and I was going to work on NEF, and NEF then and for the subsequent years, until quite recently actually, became known to me and I guess many other people as the postdoc graveyard. Okay? It was really resistant to any insight. But Bryan had a grant to work on it, and it seemed like a reasonable thing to try. But actually, between the time I'd made the decision to go there and arriving there, the co-receptors, the HIV co-receptors, were discovered. And so the first thing I worked on was basically trying to decipher the functional anatomy of the human CCR5, basically trying to make chimeras between human and mouse CCR5 to map the domains that were functionally important, trying to figure out whether the signaling properties of CCR5 were important for its receptor activity, and they weren't. And so we had a nice, about a year and a half of work basically characterizing co-receptors. And then after that, I moved on to try and work on transcription, on the HIV-1 TAT function. And at that point, the key cofactors for TAT transcriptional activation had not been identified. And actually, in fact, one of the papers that drew me to Bryan's lab was on that very topic. It's a whole set of genetic experiments that make beautiful predictions about the nature of the TAT cofactors, that there would be a preformed protein complex that would recognize particular elements in the TAR sequence. And so about halfway through my postdoc, I switched completely from working on co-receptors to trying to chase TAT cofactors. Spent about 6 months trying to do that, and we were beaten to the punch by another group who identified the PTEF-B components as the TAT cofactors. And so for the remainder of my time, I worked on sort of mechanistic details of TAT transcription, which was fine. We published some nice work on that, I think.
So these decisions on what to work on, did you discuss them with Bryan? Were they mostly driven by you? How would you characterize it?
I think Bryan saw in me a diminishing enthusiasm for work on co-receptors. And I was always interested in what my colleagues were working on. There was work on nuclear transport going on at that time. This was sort of a big question that needed to be solved. And, I'd had a relatively successful first half of my postdoc, and it was a good time to take some risks. I remember actually we went out for lunch and had sushi and talked about what I could do for the remainder of my time. And after lunch, decided this was what I would do.
Is that because of the sushi?
No, I actually think it was the first time I'd ever had sushi. I wasn't that enamored with it to begin with. But I've developed a taste for it now. But,
so by the time you arrived, the lab was bigger then, right?
Yeah, I think we're about 8 or 10 people by then. And it's been that way for quite a while, right?
Yeah, it's gotten a little smaller again now, but it was about 8 to 10.
And
that was what I thought was a good size, because if you get more than about 8 or 10 people, you start to lose people somehow. People who are less successful, who are shyer and don't come and see you, and then you don't really want to go see them because they're not doing so well, and it's kind of difficult, So you get into this mutual ignore each other thing if you've got too big a lab. But if you have only 8 to 10, then you can't afford to let anybody go off the rails. You have to pay attention and try to help them through any difficulties they have.
Now, Paul, you had mentioned earlier that he was no longer doing experiments with the exception of pipetting plasmids, right? But did you use him to troubleshoot any problems that you would have? Was he still good at that?
I would, bounce things off him. But I've learned, I think probably more so since than I appreciated at the time. If someone comes up to you, or someone in your lab, and says, well, my restriction digest didn't work, there's not really a lot your mentor can do except bombard you with questions about what idiotic mistakes you made. And so I think I learned quite quickly that wasn't very productive, and I'd have to figure those things out for myself. We did have discussions, quite a lot of discussions, and Bryan had a lot of input in what types of experiments to do. We'd have weekly meetings. I think I quickly learned to get on the same wavelength as Bryan, and he influenced the way I think about these sort of things. So in the beginning, I depended on him a lot. I think by the end, I sort of absorbed a lot of his approaches.
Now, Mike said he saw Bryan often. Did you also see him very often?
Yeah, so we'd have these weekly meetings where, I made sure I had a piece of data I hadn't shown him yet to take to the meeting. But probably, every 2 or 3 days, he'd wander into the lab and say, how's it going? Sort of, it was always a little bit ambiguous as to whether it's how are you or what have you done recently. How's it going can be quite a useful phrase in that.
Do you use that in your laboratory nowadays? Someone said
You better ask other people. I try to be clearer about those things, but sometimes I'm not.
Bryan, what did you mean? How's it going?
Exactly what he said. You don't want to say what have you done in the last 8 hours? That's a little too unsubtle. On the other hand, you do want to sort of emphasize that you're still very interested in how the project is moving, and what have you done for me recently, kind of thing. But, the other thing is if you try to stimulate a discussion, and hopefully if there's some issue that somebody's been thinking about, that'll be an opportunity for them to bring it up, and you can see if you have anything intelligent to say on the topic. One thing I find is that there's a tendency for postdocs or grad students to, if you say, if you talk to them, and you agree that they're going to take a particular approach, sometimes they can be ridiculously stubborn about it, right? My philosophy is, if you can't go through a wall, walk around it, right? You've got to, if one approach doesn't work, try to think of a different approach. Don't just keep beating on the same technological roadblock. Because, the point of the exercise is get to the goal, not get style points for how you get there. Kind of thing.
So, Paul, did you have disagreements with Bryan?
I actually don't remember any particular disagreements. As Bryan said, I was kind of meek and mild at those in those days. I wouldn't have had the chutzpah to disagree with Bryan. We used to have scientific debates, but they were always about the nuances and about points that were genuinely debatable, and about which there probably wasn't one right answer. reporter assays versus direct RNA measurement. You couldn't come to an agreement on that.
So, Mike, looking back, are you happy that you did a postdoc with Bryan?
Oh yeah, it was a fantastic opportunity, and well, I only went for 2 years, but I was enjoying it so much I stayed for 5 actually. And it was an incredible opportunity, and I learned a vast amount from Bryan, and he was always it's not always a reputation he has, but he was actually a tremendous supporter of my own career then and subsequently. He was always really helpful, and he gave me some very useful advice about various aspects of how to run a scientific career. I mean, 2 of them, which I think are invaluable things, he told me. One was, he said, for God's sake, whatever you work on, make sure you've got a good assay to measure it, which I think is very good advice. And don't work on anything with less than a 2-fold effect. Which I think is all good advice.
Great.
So, no, it was a terrific experience, and we've always enjoyed each other's company ever since.
So when you left the lab, did you take projects with you?
I did to a degree. I spent most of my postdoc working on Rev, as I mentioned previously. And I did continue working on Rev for a little while in my own lab, not a project that competed with Bryan actually. But then, as he told me during my last few days, he goes, I'm not actually going to ask you what you work on because you should just develop your own ideas. But what you'll find is that within a few months, our thought processes will have diverged and there won't be any too much overlap, which is largely true, I think.
So you've never competed in any way?
Well, a little bit over the years, but we've worked on some similar things, but not in a particularly detrimental way. No.
Do you think that was a conscious decision, or just the way it turned out?
It's just the way it turns out. I just think there's so much to work on. I mean, when you're working together, of course, your thought process tend to work in parallel to a degree. And as soon as you're away in a different environment, I mean, biology just gets more and more complicated and bigger as time goes by. So, yeah.
Paul, are you happy you did a postdoc with Bryan?
Oh yes, it was career altering for me. I learned not just about how to do particular techniques and experiments, but really how to think about approaches to particular problems. What Bryan just mentioned about not trying to go through a wall but around it, that absolutely rings true to my experience, and I've tried to do the same since. You see so many people banging their head against a brick wall for years and years. And think, why? I mean, if that, even to the extent, if that problem's too hard, just work on a different problem. So, yeah, I was only in Bryan's lab for 3 years, but they were I feel it was moving from my PhD lab to Bryan's lab was sort of moving into the big leagues. That's how I felt about it at the time, and I still think that's true.
So both of you have said that you learn things that stay with you today in your labs, and of course, I think that's a great testimony to a mentor, right? And probably you didn't know it at the time, Bryan.
No, of course, you never know how people. I was very confident that these two would go on to be very successful, but of course, the proof of the pudding, etc. So it's. They've. I thought they were going to do really well, and they've done even better than I thought they would do. Which and there are now two superstars of retrovirology, and what else can you say?
So, looking at their careers, do you take any credit for that at all?
I think I'll take the fifth on that one. I mean, obviously, I think that they both learned some things from being in my lab, and I can see that sometimes in their papers. That stylistically, how they frame the question and pursue it. I can recognize a little bit of how it's the way I would have done it if I was them, kind of thing. So, and that may be coincidence, or it may be that I get a little bit of credit for that. But everybody's a function of what the process they went through, and I was a function of the time I spent at the Roche Institute and working with incredible scientists like Grace Chu and Ann Skalka and Aaron Shatkin and others, and those are the lessons you bring with you, and that's what influences you through your career.
Paul, I want to ask you the same question I asked Mike, which is, when you left, did you take projects with you?
Yes. So the project had sort of come to a natural transition point. I'd spent some time working on co-receptors, some time working on transcription, and we knew that those were the two major blocks why HIV couldn't replicate in a mouse. So in the last few months in Bryan's lab, I was trying to engineer mouse cells to support HIV replication that would have obvious impact in terms of making a small animal model. So I think Bryan came into the lab one day as I was struggling with these experiments, and he said, I don't think I want to work on that. You can do that in your own lab if you want to. Okay. But the first paper I had as an independent investigator is actually a 2-author paper. It was basically myself and Bryan. I did about half the work in Bryan's lab and about half the work in my own lab. But what Mike said about, I felt during the time I was in Bryan's lab, I had sort of trained my thought processes to be a bit like Bryan's. Certainly, I learned to write like him, and you could almost think Bryan had successfully cloned himself in a sense. But it's true, once you get into a different environment, just have different people around you and start thinking your own thoughts and don't discuss them with a particular person, then I think probably within a few weeks, our thought processes are in different directions. So people sweat a lot about taking projects with them when they leave a lab. I honestly don't think it's that big a deal. If you're an original thinker, you'll find things to do in your own lab.
I agree with that, but many people have troubles, as you know, with students or postdocs taking projects, which is unfortunate. Have either of you sent postdocs or students to Bryan to do a postdoc? Mike?
I haven't. I have suggested to a couple of students, but they haven't taken up the suggestion.
Okay.
No.
About you, Paul?
No. So once people have spent some time in New York, Durham, North Carolina doesn't seem that attractive, I'm afraid. When I showed up in Bryan's lab, I hadn't interviewed there, and you show up at Durham city limits. There's a sign that says Durham city limits, and you're in the middle of a forest. So Durham's got a bit more cosmopolitan since Mike and I were there, but it still doesn't attract quite the same type of person, I think, as somewhere like New York City does.
But there are, you can imagine there are some people in New York who would like to be in Durham. But it's interesting that you haven't received postdocs from them.
No, I think it's really annoying, actually. But I mean, I'm not unaware of the fact that I have a slightly scary reputation. So they are in a particularly good position to tell people that it isn't totally true,
I'll say it publicly. Bryan's a very nice guy. If you're a student, I would absolutely recommend you go do a postdoc in Bryan's lab. One thing I would point out, though, is that Mike populated a significant fraction of his department with postdocs from my lab. So there has been some networking going on. Still waiting for my finder's fee for that.
Bryan, did you give a keynote yet, or are you giving it?
No, I did. I did, yes.
And what did you talk about?
Well, about the first quarter was historical and mentioned all three of the people sitting here and some others, and trying to sort of set the scene as to why I got invited to give a keynote, I guess. And then the last 2/3 were new work, which is part of which is published and a lot of which is not published yet. That deals directly with lentiviral gene regulation, which is still something I work on, but a whole new facet, which is this issue of covalent modifications of single nucleotides on RNA called epitranscriptomic regulation, which turns out to be, I think, perhaps more important than anybody had anticipated it would be in the context of the lentiviral life cycle.
That's what you talked about in Rotterdam, right?
Yeah, I did. I did a slightly abbreviated version of it at Rotterdam. We've got actually significantly more data since then, and I also had a lot longer talk.
So would you say that the majority of work in your lab is epitranscriptomics or something else?
I would say that, basically, 2/3 of the people I have in my lab work on epitranscriptomics, and 1/3 are working on an integrase project, which I haven't published on or talked about yet, which you just kind of fell into, one of these things where you wake up in the middle of the night and think, my God, what about this question? And you run into work and tell somebody to go do that, see if something good happens, right? And it turned out that it was a good idea, and so we're plowing away. But bottom line is that we've been able to generate a mutant HIV1 that's totally lacking in integrase function that is fully replication competent.
But you haven't talked about that.
And I'm not going to tell you how we did it.
All right. You come to this meeting every year?
No, I don't. I used to come religiously when I was at Roche. I think the first time I was here was in 1984, and I think I talked about the transcriptional interference story back then. I remember being as nervous as all hell. It was down in Blackfoot giving that talk. And then I think I kept going here until, I met Paul here, so that must have been like 96. So I kept every year till about 98, 99 maybe. And then I got into, I got the idea that microRNAs might be really important. But then unfortunately it turned out there were no microRNAs in HIV1, nor was HIV1 interested in microRNAs. It was totally ignored them. But it turned out that herpes viruses were made lots and lots of microRNAs. So for a while I became a quasi-herpes virology laboratory, really more of a microRNA laboratory. So at that point, I stopped showing up, and I think I disappeared from everybody's memory for a while. But we've revisited lentiviruses and retroviruses over the last few years, and certainly I much prefer working with lentiviruses because herpes viruses, 200,000 bases, they're just impossible to make a mutant is a major endeavor.
GC rich also, right? GC rich also.
Well, for a herpes simplex virus, even just PCRing out something is a bit of a struggle sometimes. EBV and KSHV are okay.
Okay. All right, let me ask you a couple of interesting questions. First, what would you say is your most significant contribution to science?
The paper that I did with Mike Malim that identified Rev as the very first nuclear RNA export protein, and identified the Rev response element, and then also the paper that we had very closely related next year, which identified what we call the activation domain, which identified what became the canonical nuclear export signal for proteins, the leucine motif.
You remember that?
How could I forget it? In fact, there was one interesting anecdote for the second paper, which was, I think I was doing the experiments for that over a Christmas holiday, and we had done a scanning mutagenesis course Rev to look for important domains and motifs. And after we'd done that, we took the ones that were negative to look whether they were inhibitors of wild-type protein to see whether they were dominant repressors. And one of them was there was a mutation in the export domain, as Bryan just mentioned. And it was a day he wasn't in the lab, so I called him at home. And yeah, I can remember this now. I don't know whether you can, Bryan. First thing was, "You're kidding me! You're kidding me!" And then it was, I'm sitting here on my bed in my underpants. An image I wasn't particularly enjoying.
I actually remember this quite vividly. It was Christmas Eve, and we had a young kid at that point. My daughter was like 2 years old, so my wife and I would take alternate alternate days on I think it was a Saturday morning. We'd take alternate days to sleep in and get up with our daughter. And it was my day to sleep in. And of course, the bugger calls me at 8 o'clock in the morning, and I was like, "Rawr." And I was, oh wow, because I mean, it would obviously something that to get a dominant negative, which at that point there was no such thing as a dominant negative for any HIV-1 protein, and I knew that was going to be dynamite and would lead to so many insights, and not to mention a nice Cell paper which came out very soon afterwards.
All right. If you had not become a scientist, what would you have done?
I could have been a businessman. I mean, I'm very organized and very punctilious. As Paul said, I deal with things immediately. I do everything in what I call it's not my term, but I do everything on the Ohio principle. I sort of only handle it once. So I either do it immediately or throw it out. Which means that, people come into my office, now these guys can all testify to this. It's empty. My desk is empty. There's nothing on it. That's because, I come in, do it, throw it out. It's one or the other. And I think that kind of organizational skill would stand me in good stead in a business environment, but it wouldn't be anywhere near as interesting.
You always used to get at me at how messy my bench was.
That's because your bench was really messy.
I didn't have time to tidy it.
Well, did he threaten to take the bench away from you? Because in my time, we would always say, if your bench is messy, you clearly don't need it, so you don't need it.
And I still say that. You get these people, they have like 2 square feet in front of them that's empty, and the rest of it is garbage. So you obviously only need 2 square feet of bench. I mean, take the rest away.
Bryan, RNA or DNA?
We've worked on a lot of things over the years, anything that looked like an opportunity, but I always really liked working on post-transcriptional gene regulation. That's always been my focus, whether it was, TAT working on the TAR RNA, Rev working on the RRE, and now we're doing epitranscriptomics, a lot of work that we did on microRNAs. So that's been the overriding theme of my research over the years, has been RNA sequence-mediated gene regulation, so RNA.
Mike, how about you? RNA or DNA?
Well, my career the aspects of HIV I worked on since Brian's time with Brian. So I've gone more towards DNA, actually, I have to confess.
Paul?
DNA is just information. RNA actually does stuff.
What's your answer?
RNA.
What's your answer? RNA? Ann, RNA or DNA?
DNA.
All right, one more question for Bryan, and then we'll wrap up. If you could have dinner with anybody, living or dead, who would it be?
You should give me a little bit of warning on that kind of thing.
No. You can think about it while I wrap up if you'd like.
All right, let's do that.
All right, so this is TWiV. You can find it at microbe.tv/TWiV. Questions and comments, TWiV@microbe.tv. And if you really like what we do, consider supporting us financially, because that's how we do these shows. We don't have very much other support. You can go to microbe.tv/contribute. You could give $1 a month, less than a cup of coffee. And you get not only TWiV, but many other fine science podcasts. Science, not science fiction. My guests today have been from Duke University, Bryan Cullen. Thank you for joining us.
Thank you.
From the from Fox Chase Cancer Center, Ann Skalka. Thank you, Ann.
You're welcome.
From Rockefeller University, Paul Bieniasz. Thank you, Paul.
Thank you. I enjoyed it.
And from King's College London, Mike Malim. Thank you, Mike.
Thank you, Vincent.
I'm Vincent Racaniello. You can find me at virology.ws. I want to thank Retroviruses 2019 for bringing TWiV back, in particular the meeting organizers, Claire Jolly and Heinrich Gottlinger. I want to thank ASM and ASV for their support of TWiV, and Ronald Jenkins for his music. And I will give you a few more minutes. In the meantime, I have these shirts that I would like to give you, but I don't think there are any TWiV fans here. Really, you don't want them, right? Who else? There's one more. These are cool. Look at the back. We have all the tour dates from last year. We'll make a new one every year. I'm not going to look at who it is. We're just going to toss it. That was a shitty toss.
Dear Peterlin.
So, Bryan, who do you want to have dinner with?
I decided to choose Arthur C. Clarke because he was one of my all-time favorite science fiction authors, and he was actually also an outstanding scientist who was instrumental in the development of radar during the Second World War. So I think he would be a really interesting character.
That's a cool choice. Yeah, very good. All right. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.